Good morning, children. Welcome back to the online classes. Yesterday we have already started with the second chapter, that is sexual reproduction in flowering plants, in which we have already studied about the parts of the flower. What is a flower first, and then we have studied different parts of a flower. We have studied about the calyx, corolla, androecium and gynoecium. Today we will start with pre-fertilization structures and events. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Primordium means the initial cells or the tissues which are being produced. Okay, so here it is floral primordium, means the cells or the tissues which are developed in the early stage, which later helps in the formation of the flower, is called the floral primordium. Okay, then inflorescences are formed which bear the floral buds and then the flowers. In the flower, the androecium, which is the male reproductive part, and gynoecium, which is the female reproductive part, differentiate and develop. Now, let us see different structures and events which are involved in pre-fertilization events. First, we will start with development of male gametophyte, okay, which involves the different things like stemming. The first is the stemming. The first thing is the stemming, right? So let us study about it. First, stemming. Now, as I have already discussed yesterday, it is the male reproductive unit of angiosperm, and it consists of two things. The first thing is the long and slender stalk, which is called the filament. Okay, it is long and slender. Slender means soft, and it is long. Okay, and the second one is the terminal, generally bilobed structure. दो लोब वाला structure होता है जिसे हम कहते हैं anther. Okay, so these are the two parts of stamen, that is filament, and the second one is the anther. Okay, the proximal end of the filament is attached to the thalamus or the petals, which is called the epipetalus. Here I have written Czech terminology. It is epipetalus. Okay, so when the filament is attached, the proximal end means the below end, the base end of the filament is attached to the thalamus, or it is attached to the petal. If it is attached to the petal, it is called epipetalus. The number and length of the stamens are variable in flowers and different species. Okay. Now, monothecous and dithecous, we will be studying it in the next slide. Let us study about the structure of anther. A typical angiosperm anther is bilobed. It is bilobed. दो लोब वाला structure होता है, with each lobe having two theca. Okay. In each lobe, see, these are the two lobes. This is first lobe, this one is the first lobe, and this is the second lobe. Right? So in each lobe there are two theca. Right? As a result, it is called dithecous. It is called dithecous. When there are two theca, it is called dithecous. If there is only one theca, it is called monothecous. Right? In last slide, you have seen that monothecous is found in hibiscus and dithecous in brinjal. Okay, so I hope you have understood what is monothecous and dithecous. If in the bilobed structure, and if in the two lobes, okay, in each lobe there are two theca, it is called dithecous, and if in each lobe there is only one theca, it is called monothecous, right? And both these lobes are separated by the longitudinal groove which runs lengthwise, okay, and in the cross section, the anther is a four-sided structure which is tetragonal. You can also say it is tetragonal in structure, consisting of four microsporangia located at the corners. Here in the diagram you can see this is one microsporangium, this is another microsporangium, this is third one, this is fourth one, right? So in the cross section means the transverse section. जब कुछ ऐसा है, right, this is bilobe structure like this, and if we cut the anther like this, it is called the transverse section or the cross section. So when we take the cross section, the anther is a four-sided, you can see 1, 2, 3 and 4, four-sided or tetragonal structure consisting of four microsporangia. This is one microsporangia, this is second one, third and fourth one. Singular is microsporangium and plural is microsporangia. S-P-O-R-A-N-G-I-A. Okay, so plural is microsporangia, which is located at the corners. Wait for a second. Okay, plural is microsporangia. Okay, so microsporangia are located at the corners, two in each lobe, right? This is one lobe, so one microsporangium, second microsporangium. So in each lobe there are two microsporangia, and later this microsporangia develop and become the pollen sac. Okay, see here you can see in this diagram, this microsporangia in the later stage of the development will get converted into pollen sacs. Okay, and in the pollen sac there are many pollen grains. Okay, or in other words you can say that pollen sacs are packed with the pollen grains. Clear? I hope there is no doubt in this. So let us move to the next slide. Okay, so now we will be studying about microsporangium. Till now, what we have learned? We have learned that anther is a bilobe structure. This is first lobe, this is second lobe. The two lobes are separated by a longitudinal groove which runs lengthwise. Okay, and in each lobe there are two microsporangia. As a result, the anther is a tetragonal structure where it has the four microsporangia at the sides. Okay, now we will be studying the structure of microsporangium. A typical microsporangium is surrounded by four walls. There are four walls in a typical microsporangium, and the four walls are epidermis, this first one. Then second is the fibrous layer which is known as endothecium. It is called endothecium. Then inner to the endothecium there are middle layers. And the innermost layer is called the tapetum layer. Okay, so these are the four layers. The first layer is the epidermis, then endothecium, then middle layers, and then the tapetum. Okay, we are talking of the structure of microsporangium. There are 4 wall layers in the microsporangium. The outermost is the epidermis. Inner to the epidermis, it is endothecium, then middle layers, and the innermost is the tapetum. The outer 3 wall layers, that is epidermis, endothecium, and middle layers, they are protective in function and help in dehiscence of anther. Dehiscence means breaking or bursting, you can say. So when the anther bursts, these 3 protective layers are very important, means they help in dehiscence, and as a result the pollen grains will be released from the anther. Whereas if we talk of the innermost layer, which is the tapetum, it nourishes the developing microspores or the pollen grains. Okay, in the center of the microsporangium, there are sporogenous tissues. We will be studying it in more detail after few minutes. Right, so this sporogenous tissues later get converted into the microspores, or you can say pollen grains. So the pollen grains, the developing pollen grains will get nourishment from the tapetum layer. Okay, and if we talk about the cells of the tapetum layer, they have the dense cytoplasm. Dense cytoplasm, and generally have more than one nucleus. I hope this is clear. Okay. Now, when the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissues occupies the center of the means center. So in the center there are group of homogeneous cells. Homogeneous means they are similar. Okay, so homogeneous cells, which are called the sporogenous tissues, are present in the center of each microsporangia when the anther is young. Okay, now let us move to the next slide. Okay, fine. This is the diagram which is given in your textbook. Again, you can see the bilobed structure. This is anther. At the 4 sides of the anther, there are microsporangia. Now, if we see the microsporangia under the microscope, right, we will find it like this, where it has 4 layers. The outermost layer is called the epidermis. Inner to the epidermis, it is endothecium, then the middle layers, and the innermost layer is the tapetum. The first 3 layers, the epidermis, endothecium, and the middle layers, their function is protection, whereas the innermost layer called the tapetum layer nourishes the pollen grains. Okay. Here in this diagram you can see in the center of the microsporangia there is sporogenous tissues. Okay. And later this sporogenous tissues, they develop into the microspores, or you can say the pollen grains. And how? We will be studying it now. Okay, so each cell of the sporogenous tissue is a potential pollen mother cell, or you can say microspore mother cell. जो भी sporogenous tissue center में present होते हैं microsporangium के, they are potential pollen mother cells. You can say PMC, pollen mother cell, or they are microspore mother cells. Okay, and this pollen mother cells in turn gives rise to microspore tetrad. They give rise to microspore tetrad. This process of formation of microspore from a pollen mother cell through the process of meiosis is called microsporogenesis. ठीक है, तो PMC या फिर पोलन मदर सेल माइक्रोस्पोर टेट्राड कैसे बनाएगा? थ्रू द meiosis process. Children, meiosis you have already studied in 11th standard. It is reductional division. Okay, it occurs in two steps, meiosis 1 and meiosis 2. Okay, so by the end of the meiosis, 4 daughter cells are produced. So here you can see microspore tetrad is produced, and this process of formation of microspore tetrad from pollen mother cell or microspore mother cell through meiosis process is known as microsporogenesis. Okay, now right, so this is all what we have discussed, right? The structure of the anther. Again, this we have already discussed. When the anther is young, a group of compactly arranged homogeneous cells, that is the sporogenous tissues, occupies the center of each microsporangium, and the cells of the sporogenous tissues undergoes meiotic division, means the meiosis, to form microspore tetrad. Okay. This is a diagram of a mature dehisced anther releasing the pollen grain. This is mature anther, and dehisced means it has been broken. The walls have been broken and it releases the pollen grains out. Right? Now this diagram shows about the microsporogenesis, that is potential pollen or potential microspore mother cell. Okay, cells of the sporogenous tissues are the potential microspore mother cells. They undergo meiosis process and it produces the microspore tetrad. Okay, microspore tetrad means 4 cells which are haploid. They stay together. When the anther matures, dehydration takes place. That means the water will be dried. Okay, and as a result there will be dissociation, means separation of these 4 microspores, which are called the pollen grains. Okay, and that is the process of microsporogenesis. Okay, now let us move to the next slide. Okay, in this slide we'll be studying about the male gametophyte, which is also known as the pollen grain. Okay, so this is about male gametophyte. Or pollen grain. Okay, so pollen grain is a haploid, uninucleate and minute spore produced in large number by meiosis in the microspore mother cells. Okay, haploid means it has only one set of chromosomes. Uninucleate means it has only one nucleus, and they are minute spores which are produced in large numbers by the meiosis process in the microspore mother cells. Okay, and they vary in their size, shape, color, design from species to species. Pollen grains are generally spherical. Here you can see the structure, okay, their shape. They are spherical. They are generally spherical, measuring about 25 to 50 micrometer in diameter. 25 to 50 micrometer. in diameter. It has a two-layer wall, which are also known as sporoderm. The wall is also known as sporoderm. The outer hard layer is known as exine, which is made up of sporopollenin. Okay, so outer wall is the hard layer. Okay, it is thick and hard ornamental, and it is hard because of the presence of sporopollenin. Okay, and the inner layer is known as intine. Intine layer is thin and it is chiefly composed of cellulose and pectin. Okay, if we talk more about the exine layer, it is the most resistant. It is the resistant layer because of, it is hard, and it can withstand the high temperature and strong acids and alkali because of presence of most resistant organic material, which is called the sporopollenin. Okay, and no enzyme can degrade the sporopollenin. The pollen are well preserved as the fossils, and pollen grains have prominent distal apertures. Okay, they have circular apertures in the exine layer where sporopollenin is absent. Okay, and from this the pollen tube germinate, that is known as the germ pore. ठीक है, तो एक्साइन लेयर में जहां पर स्पोरोपोलेनिन एब्सेंट है, ओके, उस पार्ट को क्या कहेंगे, जर्म पोर. और वो जर्म पोर में से जो पोलन ट्यूब है वो इमर्ज होके बाहर निकलेगा. So here you can see in this diagram, this is the exine layer, the outermost layer, exine, right? This is intine, the inner layer, and exine layer, as we have already studied, it has sporopollenin. इसकी वजह से वो हार्ड होता है और वो हाई टेम्परेचर, स्ट्रॉन्ग एसिड्स और एल्कली को विथस्टैंड कर सकता है और कोई भी एंजाइम उसको डिग्रेड नहीं कर सकता. ठीक है, तो एक्साइन लेयर में कहीं कहीं जगह पर स्पोरोपोलेनिन एब्सेंट होता है और उस पार्ट को हम कहेंगे जर्म पोर. और जो इंटाइन लेयर है, फ्यूचर में ये जर्म पोर में से पोलन ट्यूब को जर्मिनेट करेगा. जो इंटाइन लेयर है, इनर लेयर है, इट इज मेड अप ऑफ सेल्यूलोज एंड पेक्टिन. Again, this is a diagram of the layers of the pollen grain, where you can see this is the outermost layer which is called the exine. ये exine layer है, it is already labeled here. ठीक है, exine layer में क्या होता है, स्पोरोपोलेनिन होता है. जहाँ पर स्पोरोपोलेनिन एब्सेंट है उसे हम क्या कहेंगे, जर्म पोर कहेंगे. और उसका अंदर का जो लेयर है, दैट इज इंटाइन लेयर, एंड इट इज कंपोज्ड ऑफ सेल्यूलोज, हेमीसेल्यूलोज एंड पेक्टिन. ओके, okay, the cytoplasm of pollen grain is surrounded by a plasma membrane. Mature pollen grain inside has intine and exine. Okay, and during the development it forms microspore. From microspore mitosis, two cells are produced. Okay, let me explain this in detail. A pollen grain is partly germinated microspore representing the male gametophyte. Each mature pollen grain in angiosperm has two cells. ठीक है, जब ये पोलन ग्रेन जर्मिनेट करेगा, वो माइटोसिस सेल डिवीजन करेगा, तो वो क्या करेगा, इट विल प्रोड्यूस द टू सेल्स. इट इज गोइंग टू प्रोड्यूस टू सेल्स, and these two cells are the vegetative cells, which are also known as tube cells, and the generative cell. Okay, so the vegetative cell is larger, has abundant reserve food material, and a large nucleus which is irregular in shape. Whereas if we talk of generative cell is small. It is spindle shaped with the dense cytoplasm, and it floats in the cytoplasm of the vegetative cell. ठीक है, so जब ये पोलन ग्रेन का न्यूक्लियस mitosis करेगा, then it will result in formation of the two cells. The one is vegetative cell and the other one is the generative cell. Vegetative cell कैसा है? Large है, उसके पास abundant food reserve है, and it has nucleus which is irregular in shape, and the generative cell is small in size and it has the spindle shape. It is of a spindle shape and it has dense cytoplasm, and it floats in the cytoplasm of the vegetative cell. Okay, now this few points we have already discussed, that the pollen grains represent the male gametophyte. Their sizes, shape, colors and designs varies from species to species, and as they can remain for a long period of time, they are also considered as the fossils, right? Because of presence of sporopollenins. They can be preserved by method known as cryopreservation, which is used in crop breeding. Okay, it can be preserved at minus 196 degrees Celsius. Now, pollen grains are generally shed at the 2-cell stage in over 60% of angiosperms. In the remaining 40% of the species, the generative cell divides mitotically and give rise to 2 male gametes before pollen grains are shed. That is 3-cell stage. ठीक है, तो इसका मतलब क्या है कि जब पोलन ग्रेन्स 60% ऑफ द एंजियोस्पर्म्स में पोलन ग्रेन्स कैसे शेड होते हैं? जब 2-सेल स्टेज में होते हैं, मीन्स उसमें वेजिटेटिव सेल होता है और जनरेटिव सेल होता है, तब वो शेड हो जाते हैं. But in 40% of the angiosperms, what happens? The generative cells again divides mitotically and produces the male gametes. तो उस टाइम पर वो शेड होंगे जब 2 मेल गैमेट और 1 वेजिटेटिव सेल है पोलन ग्रेन्स में. Pollen grains of several plant species may cause severe allergies and bronchial infections leading to asthma, okay, and the bronchital diseases, right? Or bronchial infections, you can say, leading to asthma, bronchitis. Example, Parthenium or carrot grass came to India as a contaminant with imported wheat, and it became ubiquitous. Ubiquitous means it was present everywhere in occurrence and caused the pollen allergy. Now, pollen grains are rich in nutrients. So, in Western countries, large number of pollen products in the form of tablets and syrups are used to increase the performance of athletes and race horses. Then, viability of the pollen grains depend on temperature and humidity. It varies from species to species. Example, 30 minutes in case of wheat, rice, and several months as in some members of Solanaceae, Rosaceae, Leguminosae, etc. And once the pollen grains are shed, they have to land on the stigma before they lose their viability if they are to bring about fertilization. Okay, if they are not reaching to the stigma before they lose the viability, then there will be no fertilization taking place in the plants, and as a result there will be no fruit formation, no seed formation, and no new plant can be developed. Right? So before the pollen grains lose their viability, they have to reach to the stigma. So that is all for today. In our next video, we will be continuing with the new topic, that is development of the female gametophyte. I hope this much is clear to everyone, and I insist you to read the topics which we have completed today. Thank you.